Hello everyone, and welcome to Biopedia, a podcast where we examine biological concepts, sources, or people one at a time. This week, we are going to dive straight into the world of microbiology by discussing biofilms, a source of great irritation for dentists and doctors alike. So, what is a biofilm exactly? Well, it's a collection of bacteria or microorganisms encased and surrounded by extracellular material. Think of it as a bit like a chocolate chip cookie. The bacteria are the chips, and the surrounding material is the dough. All biofilms contain an extracellular matrix, which is frequently composed of polysaccharides, as well as other components such as DNA and proteins. Its foremost role is to protect the bacteria inside it. Bacteria which are floating around, or planktonic bacteria, are exposed to the full wrath of the immune system, while these stationary, sessile bacteria surrounded by the biofilm. This can lead to immune cells being unable to use their antimicrobial secretions against the microorganisms, but instead these same molecules end up damaging the body's own cells. This is a phenomenon known as frustrated phagocytosis. The creation of a biofilm is dependent on something called quorum sensing, which is essentially the ability of a bacterium to sense population density. In fact, a molecule known as BDSF can cause the bacterium Pseudomonas aeruginosa to downregulate its quorum sensing, meaning that it becomes more tolerant of a high population density, such as that needed for a biofilm. Some bacteria, when given the choice, if you like, between a regular carbon source and a social one derived from the end product of another species' metabolism, will choose the social carbon source, which can have implications in later processes. This nutritional cooperation also happens in the 5 species found in plaque, which degrade a particular kind of salivary glycoprotein, to the great irritation of dentists, I'm sure. In another example, the bacterium Agregibacter actinomycetum comitans was shown to be more pathogenic and better able to colonize a mouse abscess when it was cultured alongside Streptococcus gordoni, which seems to indicate that species choose to form biofilms in cases such as these when there is an obvious benefit to doing so. The formation of a biofilm may be a response to antibiotics. When bacteria first sense antibiotics, they may begin to form a biofilm in order to protect themselves, at a concentration when the antibiotic can be sensed but is not yet fatal. In fact, the subject of our next episode is another bacterial response to antibiotics, which can also take place in a biofilm, bacterial persistence, which can be responsible for infections reappearing even after antibiotic treatment. But as I say, that is a discussion I will have to save for a future episode. In the meantime, feel free to get in touch with the podcast at biopediapodcast@gmail.com. That again, biopediapodcast@gmail.com, whether it's to suggest a future topic, make a correction, say what you thought of the level of detail, or simply to say hello. Sources will be in the episode description by the time this is released. Thank you very much for listening, and until next time, have a great week.